Okay, in today's lecture we will discuss about animal cell culture, their origin and principles of cell culturing. First of all, a bit introduction about the tissue culture. Tissue culture is basically the general term which is used for the removal of cell tissue and organ from the animal and their subsequent placement into an artificial environment which is suitable for their growth, and then cells which after growth were removed from the organ fragment separated in single cell condition or placed in suitable culture medium, then it is called as animal cell culture. History of animal cell culture is about 100 years old when in 1907 Harrison for the first time tried to culture nerve tissues from a frog embryo in serum which was extracted from the blood clot. In 1912, Carroll attempted to study the behavior of fragments of the heart of chick embryo. These experiments laid the foundation of animal and tissue culture technology. In 1946, Bach and his colleagues used rodent tissues to produce continuous cell lines. Cell lines established in this way generally die after 50 generations. In case of human cell cultures, chicken cells have even more shorter span of population doublings. Immature cell lines are derived from malignant tissues, and the famous example of which is HeLa tissues. However, cells of mammalian origin have to be subcultured in order to maintain the continuous cell lines. There are a number of established cell lines that have been made to survive for a longer period of time. That includes 66 cells of mouse embryos, Chinese hamster ovary, etc. These cell lines survive prolonged period if subcultured at regular intervals. Alex Isaac in 1966 reported the production of some antiviral substances in the cells of many vertebrates after viral infections. Isaac named these substances as interferons as they interfered the entry of viruses in uninfected cells and inhibited the proliferation cycle of viruses. The cell culture have been used for decades for propagation of viruses for vaccine preparations. Interest has been furthered by exploiting viruses to produce specific proteins and monoclonal bodies. There are three different types of interferons. These include alpha interferons, beta and gamma interferons, each one having antigenic properties. Since 1980s, interferons were used for genetic engineering techniques. Another product which is very important for is the protein called the erythropoietin, which has put the biotechnology industry on a high pedestal. This protein stimulates the cells in bone marrow to differentiate into blood cells. This is being manufactured by recombinant DNA technology. In addition to these products, insulin, human growth hormones, enzymes, these are all produced on large scale by this technique. Another area in which cell culture technology has been successful is the somatic cell genetics, which is an area which treats the genetic disorders in unborn fetus. Transformation of cells is successfully carried out in producing transgenic animals and chimeras. Stem cell technology has proven to be a boon to a medical science for gene replacement therapy. The pluripotent stem cells can be genetically engineered without altering their pluripotency. Basic steps which are involved in the primary cell cultures. First step is basically the isolation of cell. In this step, cells are isolated from particular tissues. Then disintegration or disaggregation of cells. This can be done through chemical or mechanical methods. In chemical methods, different type of enzymes such as trypsin, collagenase are used, while in mechanical method, different type of tissue homogenizers are used to disaggregate the cells from the parent cell. Then incubation of the cell in a suitable culture media is very important. Then after many cells lost their viability, but many of them start to grow and divide on new cells. If the cells multiply repeatedly, grow for a long time, these cells can also then put into plastic bottles with fresh culture media. These are termed as the primary cell lines. After when the primary cell cultures has grown up and achieved a proper growth state, they are subcultured to give them more nutritional support for continuous growth. The subculturing is necessary step to maintain the cell in continuous growth phase, as after some times of primary culture consume the provided nutrients, and due to lack of nutrients, cell growth becomes stagnant and leads to the death of the cell. When the primary cell line continues to grow and divide, then they are subcultured in fresh media to maintain their viability and growth. After subculturing about 70 times, these cells are called established cell lines. Primary cell lines can grow very slowly, but established cell lines can grow faster, as there are some alterations in the cell metabolic mechanisms due to many subculturing steps. Primary cells maintains the properties of parent cells, but established cell lines has some alterations or altered properties because of the many steps which are involved in subculturing. Okay. When subsequent subcultures, which are more than 80 times subculturing established cell lines, cell becomes immortal, and normal cells have a limited cell growth, but some cells acquire a capacity of growth for indefinite time. This capacity is generated due to transformation in genetic material. Due to this transformation, cells lose the sensitivity to external stimuli and sometimes show changes in the chromosomal numbers. Here is the flow diagram which is indicating the culturing of animal cell. First of all, you will take the organ, and then tissue or cell fragment will be isolated from that organ. Then enzyme digestion will be carried out to get the single cell, and then this single cell is cultured on suitable culture media. Then after the culturing of the suitable media, this is known as the primary cell culture. Frequent subculturing, around 70 to 80 times of primary culturing in suitable media, leads to the formation of established cell culture. And after more than 80 subculturing, there is alteration in cell division and characteristics, so chromosomal and chromosomal number, and cells acquires indefinite growth properties. So in that case, these cells are known as the transformed cell cultures.